Here's a question for you. If you watched a stranger share their lunch with someone every day for a week, do you think you'd become a more generous person yourself, even without anyone telling you to?
Oh, that's a good one. I'd like to say yes, but I genuinely don't know if that's how it actually works in the brain.
A new study published in Nature Neuroscience just answered exactly that question in mice. And the answer turned out to be yes, and the researchers pinpointed the specific brain region that makes it happen. Let's set the scene. The ability to learn from watching others is called observational learning, and it's one of the most powerful tools social animals have. Think of a child watching a parent cook. They absorb skills without ever touching the stove themselves.
Right, like learning the rules of a game by watching before you play.
Exactly. Now, scientists have studied observational learning in animals quite a bit, but almost all of that work has focused on learning about danger, watching another animal get frightened by a predator and then being afraid yourself. That's called observational fear learning.
So we know how watching others teaches us to be afraid, but not how it teaches us to be kind.
Sharp observation. That gap is precisely what this study targets. Prosocial behavior, meaning actions that benefit others, like sharing food, is fundamental to how social groups function. But the neuroscience of how animals learn to be prosocial through watching, rather than through their own direct experience, was almost completely unknown.
And that seems like a pretty important thing to understand, especially when you think about how children pick up social behaviors by watching the adults around them.
Absolutely. The researchers at the University of Milan, led by Diego Scheggia, built a mouse model specifically to study this. They wanted to know two things. Can mice actually learn to share food just by watching another mouse do it? And if so, which part of the brain makes that learning possible? The central idea of this paper revolves around what the researchers call the social decision-making paradigm. Let me walk you through the setup, because it's genuinely clever.
Please, I'm picturing tiny mice in a little decision booth.
Not far off, actually. Picture a behavior testing box with two nose poke holes on one wall. A nose poke is exactly what it sounds like. The mouse pushes its nose into a small hole to trigger a food reward. One hole delivers food only to the mouse making the choice. That's the selfish option. The other hole delivers food to both that mouse and a partner mouse in an adjacent compartment. That's the prosocial option.
So the mouse has to genuinely choose between getting something just for itself or sharing with a neighbor.
Precisely. Now here's the key twist. Each decision-maker mouse, called the demonstrator, was paired with an observer mouse in the next compartment, separated only by a wire mesh. The observer could see, smell, and interact through the mesh, but could not make any choices themselves. They just watched for 5 days.
So they're essentially in school, watching a classmate solve problems before getting their own turn.
Great analogy. Then, after those 5 observation days, the roles switched. The observer was moved into the decision-maker's position and allowed to start choosing. The critical question was, would the observer have learned anything useful from all that watching?
And I'm guessing the answer is yes. Otherwise, we wouldn't be doing a podcast about it.
The answer was a very clear yes, and the details of how or what make the findings remarkable. A key measurement the researchers used is something they call a coherence index, essentially a score of how consistently a mouse sticks to one choice across sessions. High coherence means the mouse has a clear, stable preference. A score near 0 means it's essentially guessing randomly between the two holes. So how did the researchers actually test and control all of this? The behavioral work involved 56 demonstrator-observer pairs, and the team used several clever comparisons to make sure they were measuring real learning rather than a simpler effect.
Like what kind of simpler effect?
Good question. One concern is that maybe the observer is just picking up smells or sounds from reward delivery and responding to those, rather than actually watching and learning. To rule this out, they ran a control group where an opaque partition replaced the wire mesh, so the observer could hear and smell everything but couldn't see the demonstrator at all.
And that group presumably didn't learn.
Correct. Those observers performed at chance level throughout, no learning. Only the mice that could visually watch their demonstrator developed a clear, stable preference on day one of the decision-making phase. That's a striking contrast.
Wait, day one. They already knew what to do on the very first day of their turn.
Exactly. Most demonstrator mice started their own decision-making at chance level. It took them several days of trial and error to settle on a preference. But most observer mice already showed a consistent preference on day one before they had made a single error themselves.
That's kind of astonishing. It's like being handed the answer key before the test.
A fitting way to put it. The researchers also verified that this wasn't simple imitation. When they moved the nose poke configuration to the opposite side for a subgroup of observers, those mice adapted perfectly well. They had learned the concept of prosocial versus selfish action, not a specific motor routine.
So they understood the meaning, not just the motion.
Right now, to find which brain region was responsible, the team used several neuroscience tools. First, they looked at which brain areas showed elevated activity after the observation phase using a marker called CFOS, a protein that neurons produce when they've been recently active during learning. Among several regions examined, one stood out clearly in observers compared to demonstrators: the dorsal CA1 of the hippocampus. The hippocampus is a seahorse-shaped structure deep in the brain that is central to forming memories and learning context-dependent associations. Its dorsal CA1 subdivision, the backhand top portion closest to the cortex, often abbreviated as DCA1, was the key area.
So the hippocampus is already famous for memory. Why hadn't it shown up in social learning research before?
Had appeared in research on learning about danger from others, but learning prosocial behaviors through observation was a completely new territory. The researchers then found that observation also produced structural changes in DCA1 neurons, specifically an increase in a type of dendritic spine called mushroom spines. Dendritic spines are tiny protrusions on neurons where connections between cells form, and mushroom spines are associated with strong, stable synaptic connections that mark learning. Here's where the results get really compelling. To prove that the DCA1 isn't just active during observational learning, but is actually required for it, the researchers used a technique called chemogenetics. The idea is elegant. You inject an engineered virus into the target brain region, causing neurons there to produce an artificial receptor that responds only to a specific drug. Then you give the drug before each observation session to silence those neurons and see what happens when the observer finally gets to make choices.
So it's like putting earmuffs on just that one brain region, while the mouse watches its partner.
That's a wonderful image. And when the researchers did this, silencing the DCA1 during the observation phase, observers lost the ability to develop a clear social preference. Their coherence index was significantly lower. But here's the nuance. These mice still learned how to perform the nose poke action itself. They made just as many nose pokes as normal mice. What they lost was the ability to link the action to its social consequence, who benefits from which choice.
So the DCA1 isn't about movement or general motivation. It's specifically about understanding that an action has a social meaning.
Precisely, and the region was only required during observation, not later. When the researchers instead silenced the DCA1 during the decision-making phase, after observation was already complete, there was no impairment. The learning had already been stored elsewhere. The DCA1 was critical for acquiring the social association, but not for recalling it.
That's a really clean dissociation between learning and memory.
It is. The fiber photometry recordings added another layer. Fiber photometry is a technique where you implant a tiny optical fiber into a brain region to measure the fluorescent glow of a calcium-sensitive protein called GCAR-MP that neurons produce when they fire. By tracking this glow in real time during the observation phase, the researchers could watch DCA1 activity as the observer watched its partner make choices.
They were essentially live streaming the hippocampus.
Well put. What they found was remarkable. Observers who later turned out to be prosocial, sharing food when given the chance, showed a sustained suppression of DCA1 activity lasting roughly 40 seconds every time their partner made a prosocial choice. Observers who later turned out to be selfish showed the opposite. a rapid, brief spike of activity at those same moments.
So the same event, watching the partner share food, produced completely different brain responses depending on what kind of mouse you were.
Exactly, and the magnitude of that difference in DCA1 response during observation predicted with strong reliability which behavioral category the observer would later fall into. The brain was already registering its future preference during the watching phase, days before the mouse ever made a choice itself. So what does this tell us about the underlying mechanism? The researchers propose that the DCA1 is building something like a cognitive map of the social situation, tracking which action leads to a shared reward and encoding that association in a way that guides future choices.
I've heard of cognitive maps for physical space, like how rats learn a maze. But a social cognitive map?
Exactly the extension this paper is arguing for. The hippocampus is famous for its role in spatial navigation, but researchers have increasingly suspected it also maps more abstract kinds of space, including social space, tracking who does what for whom. This study gives that idea a concrete neural and behavioral grounding.
So the hippocampus might be charting which action helps which partner the same way it charts which corridor leads where.
A vivid way to frame it. The optogenetic experiments clinched the causal story. Optogenetics is a technique where you use light to switch specific neurons on or off with millisecond precision by engineering those neurons to carry light-sensitive proteins. When the researchers inhibited DCA1 neurons precisely at the moment the demonstrator made a prosocial choice, observers became more prosocial overall. When they activated DCA1 neurons at those same moments, observers shifted toward selfish choices.
Wait, that's kind of counterintuitive. Activating the hippocampus makes the mouse less generous.
It is counterintuitive, and it points to something subtle about how neural activity encodes learning. The natural state in prosocial observers is a suppression of DCA1 activity when they witness sharing, and mimicking that suppression artificially with the inhibitory opsin pushed mice toward prosocial behavior. Artificially activating the DCA1 at those moments disrupted that natural suppression pattern and pushed mice the other way.
So the hippocampus learning to be quiet at the right moment is what lets you absorb the generosity of what you just watched.
That's a beautiful way to put it. The broader picture is that the DCA1 acts as an interface between the cognitive processing of what you observe and the motivational systems that shape what you choose to do later. The authors also note that a circuit connecting the prefrontal cortex and the basolateral amygdala, an almond-shaped structure involved in emotional and reward learning, has been linked in prior work to encoding the social value of rewards. Together, these regions may form the neural backbone of social learning, both through direct experience and through observation.
So what's the single biggest takeaway from all of this?
If I had to put today's central surprise into one sentence, it's this. The hippocampus doesn't just remember where you've been, it remembers what you watched someone else do for another person, and that quiet neural recording shapes whether you'll be generous the next time you get the chance.
That feels deeply important, especially for understanding conditions where social learning breaks down, like some neurodevelopmental or psychiatric disorders.
The authors make exactly that point. They suggest this mouse model could be valuable for studying disorders in which the ability to learn from others' actions is compromised. The research was led by Diego Scheggia and colleagues at the University of Milan, published in 2026 in Nature Neuroscience, under the title Individual Differences in Prosocial Learning are represented in the hippocampal dorsal CA1. You can find it at doi.org/10. 1038/S41593-026-02292-2.
Thank you. I'll think about the hippocampus the next time I see someone being generous in public.
That's exactly the right reaction. Thanks for listening to Oskai Pod.